Hello everyone, I'm Rayana Feltrin and I'm currently a master's student in the Laboratory of Photobiology at the Federal University of Santa Maria, located in South Brazil. And today I will be presenting to you my work entitled Occurrence of Xeroderma Pigmentosum D helicase in Galliform birds. Well, genomes are constantly vulnerable to damage coming from different sources. However, several kinds of DNA repair pathways developed throughout the evolution of species to ensure the maintenance of genome integrity. The most versatile of them is the nucleotide excision repair, NER for short, as it can remove a wide variety of bulky lesions. As you can see in this figure, the process of repair by NER occurs basically in 4 steps: damage recognition, excision of damaged nucleotides, DNA resynthesis, and strand ligation. The Xeroderma Pigmentosum D or XPD helicase integrates the transcription factor 2H and has an important role before nucleotide excision in order to allow the separation of the double helix into single strands. XPD is one of the most conserved proteins of NER, being present even in archaea. However, a canonical XPD is apparently missing in Gallus, as we concluded in a previous study, so we decided to investigate further. In order to do so, we have carried out a refined search of XPD using sequences of the American alligator as queries for tBLAST-N and BLAST-X searches against the genomes and proteomes of 15 species of Galliforms, the order of chicken, in addition to 4 species of Ctenomyforms and 1 of Struthoniforms, which are in the base of the bird phylogeny. Then, we used 4 filtering criteria to select the ortholog sequence candidates: E-value lower than 1e to the -6, highest percent identity, closest protein length to the query, and conserved protein domains obtained by using SMART and PFAM. As the outcome, our search for XPD in chicken retrieved a sequence annotated as an ATP-dependent DNA helicase DDX11, which has the same characteristic domains of XPD as you can see in XPD of humans. In fact, DDX11 is a member of the XPD family and acts as a backup for another kind of DNA repair pathway in chicken cells. Therefore, we infer that DDX11 may have an XPD function in chicken, but functional studies are strongly required to test this hypothesis. Regarding other Galliforms, we obtained hypothetical proteins in 3 species: Bambusicula thoracicus, Callipepla squamata, and Colinus virginianus, which contain the same XPD domains, indicating they may also have an XPD function. In addition, we retrieved some sequences that, in spite of having high sequence identities, present very low coverages, as in Callipepla squamata, Pavo cristatus, and Sirmaticus mikado, for example. On the other hand, we found probable occurrences of XPD in 1 species of Struthoniforms, additionally to 2 species of Ctenomyforms. And as you can see here, the Ctenomyforms, Notoprocta perdicaria and Ctenomys guttatus, have both XPD canonical domains, with the latter also having an additional domain, HBB. In turn, Struthiocamelus is missing only the N-terminal domain. So, considering the probable occurrence of XPD in these species of Struthoniforms and Ctenomyforms, we used their sequences as queries for blast X searches against the chicken proteome. However, we did not find a canonical XPD, but the DDX11 helicase was retrieved in 2 of these searches. Taking this into account, we suggest that there might be occurred a progressive loss of the XPD sequence from the base of the bird evolution throughout the evolution of Galliforms until chicken. So, what did we find about the occurrence of XPD helicase in Galliform birds? We suggest that the DDX11 helicase may be replacing XPD function in chicken. We found that there are probable occurrences of XPD in only 3 of 20 species belonging to Cynamiforms and Struthoniforms, and we suppose that there might be occurred a progressive loss of the XPD sequence throughout the evolution of Galliforms. Therefore, our work reinforces the importance of in silico studies to open perspectives for functional investigations. I would like to thank CAPES and CNPq for the funds and the Student Council of the International Society for Computational Biology for the opportunity of presenting my work. Thank you so much for your attention.